Good morning, everybody. It's the last day of the Bioinformatics module for this week. We've been through really interesting topics, a little computer science to start us off, a lot of sequencing in the middle, and today we're going to talk about pathways and networks. Because a lot of times we have lots and lots of data, but trying to distill information out of it can be quite tricky. In a lot of cases, pathways and networks can help you get there. So I would note that the first time this subject was offered in my old class at Vanderbilt, my friend Bing Zhang put together a lot of the initial slides. So I've distorted things quite a lot over the decade since then, but I wanted you to know that he had a lot to contribute here. All right, so we're going to start by talking about gene sets, which is to say there's some sort of biological activity on the one hand and some set of genes known to be associated with it on the right. So these together are a gene set, the set of genes attributed to some particular activity. We'll talk about pathways, which have a more chain-like, network-like association to say that gene A does an operation after which gene B can do its operation. I think many of you have gone through a class where you learned about Krebs cycle. That's an example where you have this pathway of interactions. Gene Ontology is a really, really big one. Very frequently you'll just hear it called GO. We're going to talk a little bit about how gene ontology is structured. And we're also going to talk about network modules. This is a rather different way to think about genes. Instead of saying, I've got these 10 genes all associated with this one activity, we instead think about gene-to-gene relationships or protein-to-protein relationships. So this creates a network kind of topology for understanding genes and understanding what type connections can mean in biology, like feed-forward loops and so on, is something where network modules come to play. It's not just important that we project our long lists of biotechnology information into these pathways and networks, but it's also quite important that we have some sort of statistical means for assessing when a particular pathway is enriched in a result. So we're going to talk about two examples of how that can be handled, one being the over-representation analysis, or ORA, from WebGestalt. And the next one we're going to talk about is the Gene Set Enrichment Analysis. That's GSEA. Okay. This is kind of my map for how this lecture comes together. Some of these topics we're going to discuss in a lot of detail and some not so much. So I would note that a lot of work has taken place in order for us to understand activities of a large set of known genes. I think that we have the kind of classical biochemistry route that we can think about, where researchers spend their entire lives essentially plumbing the interaction of this metabolite becoming this metabolite through this interaction, through the action of this enzyme, for example. You know, people spent years, decades in some cases, untangling how these genes contributed to a particular pathway running its course. So classical biochemistry has offered us quite a lot. More recently, as gene expression data have become more broadly available, we've gained the ability to discern genes going up and down in concert, which is to say that the same particular growth circumstances lead to a rise or a fall in a particular gene's expression. So on Wednesday, I gave a talk over at UCT. It appears in the YouTube channel if you want to see that kind of details a lot of these methods. Chromatin immunoprecipitation is an approach that has been used to evaluate the association between particular genetic regions. Here you have the chromatin that are associated with a particular protein's interactions. Of course, we frequently look at which proteins, what prey proteins may be associated with a particular bait protein through protein co-immunoprecipitation. The yeast-2 hybrid system has been a really big deal for understanding whether gene products come into proximity with each other or not, although it's a little prone to false positives. And of course, there's a fair amount of work that goes on with confocal microscopy to establish the co-location of multiple proteins. And if you move into things like FRET, to be able to discern that They come into very close proximity indeed, so that the dye attached to one protein can interact with the dye attached to the other. So these are examples of the ways that we learn that genes are not acting individually, but rather the proteins are interacting in complexes. So these all result in known relationships, documented relationships. These relationships can be perceived in a lot of ways. If you're doing a point-to-point assessment, you're asking what family of genes is this particular gene associated with. So you could think of that as a point-to-point network. You could also think of a whole series of reactions that are each catalyzed by different enzymes, where each feeds into the next. That's kind of a canonical pathway approach. We'll look at examples of both. And then I would note that we have a lot of this information being captured in Gene Ontology in Go. And it's a very valuable resource about which we're going to say quite a lot. But I would note that Gene Ontology is different from some of these others in that it involves a controlled vocabulary, which is to say that there are particular terms that relate the different genes to each other. And then as we get to the subjects we'll talk about today, there are lots of ways that we can exploit the information in these pathways and networks. So network module detection is a strategy by which we recognize these close relationships among genes. Random walk assessment is a way to evaluate essentially how many steps removed from one gene is another in interactions. Cytoscape is software that we just have to talk about because it's really, really important for visualizing biological networks and gene networks. Dimensionality reduction is a big deal because frequently we have a huge number of genes that we're detecting as differential, but we would really like to understand, to reduce the size of this problem, to stop thinking about, is this gene differential, or rather, is this pathway differential? This is to reduce the dimensionality of the problem. And of course, we've already mentioned, we're going to talk about overrepresentation and gene set enrichment analysis as a way to evaluate pathways and networks that are much more common here than one would expect by random chance. So let's just start with where our data are coming from. We've already had quite a lot of talk about microarrays here with the gene chip image included. We've talked about RNA-seq, that's shown by the image below that to the left. We haven't talked about proteomics per se, but if you come to the proteomics module offered next month, is it? Is it April? Is it May? It's coming up really fast on the heels of this. You can learn all about how mass spectrometry can be used to recognize proteins that are changing. We have clustering analysis if we're trying to do data patterns discover patterns within our data. And of course, I think most people will recognize the volcano plot up there, given that we just talked about those yesterday. The information that comes from these are typically some sort of list of genes that we think are interesting. Now, how that definition comes together may change on our particular area of research, but frequently, if you just think about one cohort of cases and another cohort of controls, for example, you have some set of genes observed to differ in expression between the two. So it's lovely to have a list like this, but how do we translate that into some sort of biological inference? A lot of today's lecture is built around that concept. All right. So I would ask you at the start to think about a reconceptualization of the lists of genes or gene products that we have. We might start by saying, well, we have recognized these four proteins to be interesting: Casp, Ras, MECK1, MEK kinase 1, and MLCP. So if we imagine that these genes are really critical to whatever biological activity we're seeing here, we might go into a site like Gene Ontology or into KEGG or something and learn that Casp is known to have a MAP kinase activity, and it is also known to have an apoptosis capability. Ras is associated with a MAP kinase activity and cyclic AMP signaling. MEK kinase 1 is a MAP kinase, and it is associated with apoptosis. And finally, MLCP is associated with cyclic AMP signaling. So that's a pretty straightforward thing. In this case, we knew the genes, we went looking in a database, and back came this list of functions. But I think a lot of people find it really difficult to make the next step, which is to turn that on its head. We see that MAP kinase does not show up for just CASP or RAS, right? It shows up in 3 of the 4 genes here. So what if instead, we instead of thinking about which functions associate with these genes, we ask which functions are enriched for these genes? So here we can say MAP kinase is associated with 3 of the 4. Apoptosis, associated with 2 of the 4. Cyclic AMP signaling, associated with 2 of the 4. And already we see that in this case we've gone from 4 genes to 3 activities. So this kind of turning it over and asking what pathways do we have evidence for in our interesting genes list is very powerful in transforming how we perceive our data. The list of genes themselves may be rather undescriptive, but the enriched functions among them could be very insightful. It may help us with a lot of insights into the biology we're exploring. And my tongue is all dried out. I had a tea incident this morning. I was drinking my tea and I got close to the bottom and it started tasting kind of gritty. I learned that the tea bag had blown a hole in it. And I suddenly had a mouth of grounds. Not the way to drink your tea. All right, so tea-related injury. Here we go. Now, let us think about some of the advantages of thinking about which pathways are dominant in this data set rather than genes. First off, better interpretation. When we start with a list of interesting genes, that's lovely, but if you're trying to deduce common biological themes that arc over multiple genes, you're getting a much better way to interpret the data at hand. Improved robustness. You can guard against noise in your data. Now, what do I mean by that? Well, I would say that if you are looking at lots and lots of genes across lots and lots of experiments, you may find that there's variability in how much of this gene you're able to measure in each of these samples. As a result, you might have some genes that really were differential, but they had so much variability in them that you didn't spot it, right? So robustness is one way that we're able to reduce the extent of false positive claims, to say that this gene was differential when it's not, and to reduce false negative claims, to say that a gene did not appear to be differential when it really was. This leads to an improved sensitivity as well. It might be that for a particular pathway, 5 of the genes are expressed at a very high level, which gives you a better ability to detect that they're differential. But there may be another 5 proteins in this same pathway that are present at lower levels. It may be that if you look at these individually, gene by gene, those that were present at much lower levels were differential, and yet the statistics don't show a significant p-value for them. If you're able to consider the data as at the pathway level, it may be possible to discern that these genes really are differential, just not when you look at them individually. Right? The more data you can pull together to make your assessment of whether this pathway is differential or not, the better your ability to detect that it is or isn't. All right. Now, hopefully, there's a huge pile of these pathways out here. You can work with Biocarta or KEGG or Metabolic or Pathway Commons, Reactome, STKE, etc. There's a website out there that gives an index to all of these different resources, and it can be particularly helpful if you have an organism that may not be very well documented in one database, but is in another. I think it's called PathGuide, if I remember correctly, but it appears in the slides I gave on Wednesday over at UCT. So there are a great many of these databases to try to define what pathways look like. This is an example from KEGG. I believe this is one from Biocarta up at the top. But all of these have certain weaknesses. One is limited coverage. So it may be that we have 20,000 or so protein coding genes in human, but not all 20,000 are going to appear in any of these. There's no one database that shows all the different functional associations for human genes. In a lot of cases, we simply don't know yet, so obviously we wouldn't be able to add that to a repository. There's inconsistency among the databases. It may be that very slight differences in terminology lead to one database describing a particular functionality in a way that you recognize, and another database may represent that functionality with a different name that you didn't expect. So there's kind of this inconsistency. Different databases have different terms for the same thing. And certainly some databases have more links for a given species than do others. Often you find that these gene sets that have been described as pathways may cut things up differently. That one pathway represented in one database may have 20 genes, but that same pathway may show up in 2 or 3 pockets of genes in another database. So the relationship among these pathways is not always that well defined. By the way, that slide used to be longer, but I decided that I would actually check which of the repositories I used to list were still live. So each time that slide gets a little shorter, because a lot of every web databases in biomedicine frequently run for the first 10 years, and then they assume no one's using them anymore, and they take down the server. So it's always a good idea to check for current links on these things. Now, Gene Ontology, Go, is one of the most important pathway and network representations out there. I really feel that everybody will, at some point in their careers, make use of Go. It's very well structured, very precisely defined, and it uses a controlled vocabulary to describe the roles of genes and gene products. So one example of this is the type of relationships that may be defined within Go. We have things like is a, part of, has this part, regulates. These are terms that get defined very closely. Now, I think a lot of people see the word ontology and they come up with a blank. That doesn't seem like a familiar term to a lot of people. So I've added a definition down here at the bottom. An ontology is a theory about the nature of being or the kinds of things that have existence. So a gene ontology in this case is describing not only which genes have known functions, but how they relate to each other. So biological ontologies include some sort of controlled vocabulary and some sort of defined relationships among the genes in this case. Now, I would note that there are 3 hierarchies in one database here. It might be that you are interested in the proteasome, for example. If you're interested in the proteasome, it probably interests you a lot to know which genes are associated with the proteasome. If so, you're likely to use something like cellular component as your guide. That's not the only option, but it is a good strategy by which you can use what localization information is out there for which genes are expressed where in a cell. Biological process is also very valuable. This is not so much a biochemistry thing, but more like a molecular biology thing. There's a process that cells rely upon to do what they want to do. Those are described in biological processes. And then we can look at molecular function. This is much more like the biochemistry thing, to say which specific, what specific reactions are catalyzed by this gene, what molecular events does this mediate, etc. So gene ontology is not one hierarchy, it's 3. And each of these 3 is going to have value for very different applications. So let's see if we can talk about some of these hierarchies that we described. You could imagine that we are interested in proteins or genes relating to the plasma membrane. Okay? We see that we have extrinsic component of plasma membranes, things on the outside, intrinsic components of the plasma membrane, things that are integral and part of the membrane itself. Within this, we have the integral component of plasma membrane, the intrinsic component of the external side of the plasma membrane, and anchored components of the plasma membrane. And as we drill even deeper, we get down to things like anchored component of external side of plasma membrane, right? So you can see that higher up, we have the most abstract parts of these trees, and the lower down you get, the more specific the set of genes you're looking at. You tend to see something like a superset effect, which is to say that all genes listed in anchored component of external side of plasma membrane are also listed as part of the anchored component of plasma membranes. which means that the number of genes represented by a stage in the hierarchy is going to get higher as you go to closer to the root of the tree. The tree's hanging down in this case, sorry, by the root. I should explain that. Now, you probably noticed that there are these little I's and P's and so on. These are the relationships in this case. So we have another hierarchy over at the left. This one is starting at the very top of biological process. We see that cellular processes and single organism processes are in here. Underneath that, something that depends on both of those is the single organism cellular processes. Beneath that is cell death. And down at the bottom is programmed cell death. So I'm only showing a very narrow part of the tree here to say how does this terminal element at the bottom relate to all the layers above it. And you see that we have different little color flags on that one to specify whether they're talking about an is a relationship, a part of relationship, a regulates, positively regulates, or negatively regulates step, occurs in, capable of, or part of. So these relationships are what's used to link together these boxes. I believe in this case, the P's represent part of, and the I's represent is a relationships. So we have a hierarchy in that we have multiple levels, and we have relationships that link these boxes together. Okay. When we make use of Go, just as when we use Uniprot, we need to keep in mind that some of the relationships that have been encoded in Go are manually annotated, which is to say, somebody did some experiments, they reported it in peer-reviewed literature, and a curator of Go read that paper and said, this is a new function known for thus and so, and thus we will annotate it to be part of that hierarchy. A lot of it, however, cannot wait that long. We generate a lot of information, and being able to analyze it electronically and add electronic curations is quite valuable. So in Uniprot, we have the difference between Swissprot, which are manually curated proteins, and Tremble, which is the more electronic kind of annotation. In the same way, within Go, we have both electronic and manual annotations. All annotations must be attributed to a source, and they need to indicate what evidence was found to support this particular Go term association with a gene or protein. As a result, they've created quite a set of different codes used to describe what kind of evidence was used to get there. So IDA, IMP, IGI, IPI, and IEP all represent particular kinds of experiments that could be used to draw this association between a function and a gene. We have a fair number of computational codes reflecting that the relationship may be found by software, not by a human, and those are described as ISS, IEA, RCA, and IGC. We can go to the website and look up what all of these mean, but what if, for example, someone publishes a landmark study in which a mouse protein is found to be associated with apoptosis, one that we've never seen before? You could imagine that one computational approach might say, gosh, this gene found to be associated with apoptosis in mice has an ortholog in humans. Therefore, we're going to infer that in humans, it may also be associated with apoptosis. In a case like that, you would have an experimental code for the mouse protein, but you would have a computational code for the human protein, at least until somebody actually does the experiment in humans and says, it does the same thing here, or no, it does not do the same thing here. Author statements, sometimes the curators are actually shooting you an email to say, I saw your paper on thus and so. Does this relationship stand up in your research? And sometimes there are other codes. So you might imagine that as Gene Ontology was created in the first place, someone created an association between a gene and a function that no one remembers at this point why that association was drawn. You might have an ND code on that to reflect no data. So generally speaking, we like to track why the software, or why this hierarchy has a relationship written down. Right, so must you go through the Go website every time you want to make use of Go? Certainly not. There's a world of libraries out there to help you with that. Amigo 2 is one of the services made available by Go itself. Bioconductor, which we've mentioned in connection with R, has a really valuable connection so that if you have a list of differential genes that you found through R, you can use the mega library Bioconductor to use Go directly. And QuickGo is one of the services made available by the European Bioinformatics Institute. They have a great number of web services and web tools to support its use. So you might think to yourself, this is wonderful, I'm going to go right into Go and look up my species and find out all kinds of amazing things. If you are working in a non-model organism, you're quite likely to find that a difficult procedure because the number of annotations that are visible by species can change quite a lot. In this case, we see that Homo sapiens is very strongly evidenced. I believe that in this case, we're looking at at experimental and computational as separate colors. So we have blue and orange. So in Homo sapiens, we see that I believe less than half are known from direct experimentation because you can't do everything with a cell line, right? So we have quite a few things that we know computationally about humans that have been transferred in by other species. And Musculus is the mouse. Rattus norvegicus is the Norway rat. Does everyone know Gallus gallus? Chicken. We got the chicken up here. How about Dana rario? Zebrafish? Okay. D. melanogaster?
Sounds familiar.
It should sound familiar, that's good.
Fruit
Fruit fly, there it is. The fruit fly. The guys in my dorm wanted to get me a hat with a feather in it when I'd come back and talk about what my fruit flies had been up to that day in genetics class. So D. melanogaster is the fruit fly. What about C. elegans?
The worm.
It's the worm, right? One of the best developmental models out there. Okay, so we have some difference. I mean, you have lots of species represented here, and all of them have some amount of information there. It may be, however, that the species you're working in is not a model organism and has less information to draw upon. I think this slide is a little busy, but I think it's worth having in here because it tries to break down where the information comes from in each species in 3 different paths. So if we look at C. albicans here, anyone heard of this one? Candida albicans. I believe that's still a yeast. Okay. And we've got Oryzae, A. oryzae. That's right. Okay, how about E. coli? Have we heard of E. coli? This is good, okay. But what I want you to note about this graph is that each of them appears 3 times. Albicans 3 times, Oryzae 3 times, E. coli 3 times. A. thaliana, I think some people here have heard of A. thaliana.
Yes.
Arabidopsis, the plant, right? So each of them is showing up 3 times because it is showing where the information comes from in biological process, molecular function, and cellular component separately. So I would just point to the amount of similarity evidence we have here. Do you see that it's kind of almost a brown, a purple that tends to brown. I wouldn't have expected to use that color. It's the top part of each of these bars. Look just how tall that is across here. There are a huge number of times in which we have an experimental result from one species talking about its genes, and it's only through orthology that we transfer that information to another. So the same gene appears in multiple species through speciation. If found to do this activity in one species, we assume it does that in another species as well. Okay. Now, from here we're going to move away from Go and move on to something else that is really, really widely used called KEGG. This is the Kyoto Encyclopedia of Genes and Genomes. Everyone knows what Kyoto is?
Japan.
City in Japan. Very, very ancient, very important city in Japan. All right. So in this case, I'm going to talk about caffeine metabolism. Caffeine metabolism. We metabolize caffeine, don't we? Right? I had my tea this morning. After eating some grounds, I think I have a little caffeine in me. All right, so here's caffeine, way over here at the right. I put a little red box around it because I realize It's going to be hard to read that at a distance. What we're looking at is a roadmap of how you get to caffeine to manufacture it, and what you do with caffeine once it's in your body. So this is a digraph, which is to say a graph in which the edges have direction. So we see that caffeine may be metabolized to create something called theophylline. It may also be modified to become 1,3,7-trimethyl uric acid. Okay, so these are possible ways that we can lead away from caffeine. We could also lead to the left, to something called theobromine. But I want to point out something kind of interesting about this relationship. There are arrows pointing in both directions here. Do you see that? Which means that if you have theobromine, you might manufacture caffeine from it. If you have caffeine, you might manufacture theobromine from that. So we have both arrows here. I want to note as well that the arrows have boxes with four sets of numbers inside. You see those? Up at the top left, I've put in a little label drawn in between xanthocene and 7-methylxanthocene. That little box says 21.1.1.58. So what are these numbers? They look a little bit like IP addresses, don't they? Right? When you give the address of your computer. Well, in this case, they are Enzyme Commission codes. Enzyme Commission is really valuable information if you can get it, because Enzyme Commission numbers describe this particular chemical reaction and which enzyme is known to catalyze it. So that's really valuable information. If you see that in your species a particular activity is going on that transmutes this metabolite into that metabolite, it may be that there's already an enzyme commission code in place to represent that activity. And if you need to find an analog, you can look up all the different proteins that have been previously associated with that number. So this is something of an entry point to metabolomics, really. All right. What else do I want to say about this? I think that's basically it for now. But I would point out that there are hundreds of these maps of all these different gene relationships that have been spelled out. This is one of our first examples of a pathway being drawn out for us. Because if an organism is producing piles and piles of 7-methylxanthine, you might want to have some notion of what kind of biological processes that connect to. In my mind, I don't immediately connect 7-methylxanthine to caffeine, but they are in fact related because one may be used to build the other. All right. So in this case, we would like to talk about enriching on the basis of pathway. So here we have at the left some numbers that have _AT written on them. I think we saw some examples of that yesterday. These are probe names from a microarray. So in this case, we have a whole bunch of measurements that we've made for genes, and we want by using this microarray, and we want to learn about the pathways and networks that are represented on that list. So this is tricky because those aren't gene names, are they? _AT is a particular probe sequence on a particular microarray. So we have this translation process we need to go through. We need to start with, in this case, a probe name from a microarray and get to a gene symbol that the software understands. WebGestalt is really, really powerful because it has a tremendous ability to map information of a large number of types into entree gene IDs, which is to say gene names that NCBI recognizes. So we see that they include microarray probe identifiers from Affymetrix, from Agilent, from CodeLink, and from Illumina. So they already support out of the box a lot of the labels that would come from microarray data. It might be that you have some gene variants at hand, and it's possible to translate those from the dbSNP format into gene labels. It might be that you are working with something like proteomics data. What if you have a bunch of uniprot identifiers and you need to map those back to the genes they come from? WebGestalt is able to handle all of those different transitions between these different formats to gene labels that it can use internally. We note that at the time of this publication, I think this was the 2013 paper, that they covered 8 different organisms: human, mouse, rat, dog, fruit fly, worm, zebrafish, and yeast, which is not bad if what you're working in is a model organism. If you're doing clinical studies, yes, you can still use this. Does this work with a random soil bacterium? Probably not. Right? That's going to be a more difficult transition to work with. So these identifiers can get mapped to genes, fed into WebGestalt, and from that, the software can project against a large number of pathways and networks. We've already talked about gene ontology. You can see it gets its own box here. Biological process, molecular function, cellular component, just as we described. It speaks several different pathways: KEGG, pathway commons, wiki pathways. It has the ability to deal with network models, to deal with disease and drug associations, and even where things are located on chromosomes, where that information is available. So being able to do statistics, starting with real experimental data, is something that WebGestalt was intended to address. All right. So that lends itself to this discussion of over-representation analysis. Let me see. What if I were looking at different honors classes and trying to ask, are men overrepresented in this class? Right? So I might write down the number of men and the number of women in a given honors class, and then I could ask, are men overrepresented there? Right? So in this case, I have an expected number, because we have roughly as many men and women in our society. So I assume that an honors class should have roughly an even split between men and women. Now, at this school, in this department, we quite frequently see that women actually outnumber the men by pretty significant numbers. It was true last year as well. So I could ask them, are men overrepresented in this collection? So I'm going to get a different answer if I say, look at all of the full professors in the department. Are men overrepresented there? I could ask in the administrative staff, in the janitorial staff, in the student body, whatever. I could ask in these populations, are men overrepresented? That becomes a pretty easy example for us to understand because we have a prior associated with it. We know that in South Africa, men and women have roughly equal populations. In the same way, we might come at overrepresentation analysis in the same way to ask, is this pathway overrepresented here? But in that case, we don't have a 50-50 proposition. We might be looking at a pathway that accounts for 20 genes out of 2,000, right? That's a much smaller fraction than 50-50. So in this case, at the left, we have a development category. So we have a gene list called development. It contains 1,842 gene symbols. These are actual gene symbols rather than probe names. We have some list of differentially expressed genes that we've pulled from our microarray experiments. So our differentially expressed gene list has 581 genes on it. Some of them overlap with development, and some of them don't. So in this case, I have 581 genes on my list. I had 1,842. I'm sorry, I'm not expressing numbers well today. We have 1,842 in this particular pathway. Now we want to know how many of the genes I call differential are on this list of development. And what I see at the top is that there were 98. All right, so just under 100 genes fell on both lists. So that's like what, 1 in 6, something on that order. So is that number in the overlap an unusual, unexpected number, or is it just exactly what you'd expect? What we see is that by our statistical model, we're able to say you should have seen an overlap of 65, but what you did see was 98. So what we have is more gene hits between these two than we would expect. But that could have been just random noise, right? Just because we expected 65 by random chance. If you saw 66, would you get excited about it? Not really. because what's happened could very easily be explained by randomness. So we need to have some way that we can put a value on this coincidence and say, this is not likely to have happened by random chance, or we would have expected this to happen, say, 40% of the time. Okay, so how do you get there? I'm showing you an equation. Did anyone blanch when I showed an equation? There's a little blanching. It's okay. We're going to work our way through this. We're going to start with the contingency table. If you're doing stuff in statistics, contingency tables show up all the time. So in this case, we have some number of genes that we declared to be differential that also appear on the list of development genes. So these are hits. We have a gene that was called differential that also happens to be on this pathway. We have a set of genes that we called differential that do not appear on the list. In this case, that's represented by N minus K. It shows up in the second row, first column. We also had some number of genes that appeared on the development list that we didn't see and that we didn't call differential in our set. In this case, that's described as J minus K. And then there's a large number of genes that are neither in the list of development genes, nor are they on our list of differential genes. So that here is called M minus N minus J plus K. I realize that's a little bulky, but do you understand how all genes are accounted for with this contingency table? Yes?
Why is it G plus K?
In this case, J is the total number of genes associated with the development pathway. K was the number that are on both lists. So by subtracting all genes, the number of genes in development, by subtracting from it the number that were found on our list, we see the number that were on the development list, but not on our list of differential genes. I'm sorry, I think this slide could be a little simpler, actually. But I want you to think about this in connection with true positives, true negatives, false positives, and false negatives. It's a similar kind of thing. All outcomes are described there. So all of the total number of all genes is the sum of those 4 cells. Here we're just breaking it down. Do they appear on both lists? Do they appear only on this list? Do they appear only on this list? Do they appear on neither list? That's the set of 4 outcomes. So from that, having computed these values, we can then run this through the hypergeometric distribution. The hypergeometric distribution. So hypergeometric distribution is very, very good for analyzing tables like this. There's a story about R.A. Fisher, who created what's called the Fisher's exact test from this. He said that it's built around a story that he always called the woman drinking tea, a story of that sort. So the woman drinking tea, it was a colleague of his who said she could tell whether tea had been poured in the cup before the tea or after the tea. And he thought this was nonsense. He thought nobody got so sensitive a palate that they could tell whether the milk was added before the tea or after the tea. So he tried to think of a way that he could test her claim that she could discern which was in there. So he said, I'm going to pour you 8 cups of tea. In 4 of them, I will have poured the milk first, and in 4 of them, I will have poured the tea first. And so she did it, and I think that she got a perfect score, actually. Now, the really funny thing about this, I mentioned that some of the biostatistics people are not very nice people, right? He could have said, the scientist drinking tea, because the woman drinking tea was not some socialite he met at a party. It was a woman that he'd worked with professionally for like a decade. She had her PhD, right? So why do we just call her a woman drinking tea? Are we trying to say women make outrageous claims on no basis? That seems to be what he was trying to say. That's kind of a rude thing. So anyway, R.A. Fisher, not a nice man. That's on video. All right. So when you want to compute the probability of this particular outcome going, you can plug the numbers from this contingency table. Everyone remembers the term contingency table? Okay, well it's in the title. I probably should bold it and italicize it. So we put these numbers in here. Does everyone recognize the big sigma character? Mm-hmm. Yeah, the sum of all these things, right? So in this case, We're asking, what if we sum together this particular outcome with all the less likely outcomes as well? What that produces is a p-value, which is to say, how probable is it that by random chance alone, you would have this many hits between your differential list and this pathway, or more by random chance? Right? That's the p-value that pops out of this. Very useful test. This is the over-representation analysis, and there are lots of tools out there, including the WebGestalt one we talked about, that will compute p-values this way. Okay, so what does the feedback look like? In this case, I've actually gone to the website and run the tutorial program for over-representation analysis. We can see that the gene set that was that they were looking for, this Gene Ontology 18209, has 305 total genes in it. We provided the software with a list of 446 genes that were differential, and now it has found an overlap of 35 genes that fall in both. And so what this produced was a very low p-value that here has been multiple testing corrected by Benjamin E. Hochberg, the FDR route, producing this high enrichment ratio associated with peptidyl serine modification. So in this case, rather than just say we have this big laundry list of differential genes, we're able to say the pathway that is most heavily overrepresented in this case in our differential gene list is peptidyl serine modification. And the second highest pathway listed there is peptidyl tyrosine modification. Serine and tyrosine have quite a few things in common in how they behave. So in this case, we can get a sense of all these different pathways that have been tested against our input list of differences. Dry mouth. Okay. It is also possible to visualize these data in the context of known pathways. Now, this drawing is not one I've shown you before, but it looks really familiar, doesn't it? Just a few slides back, I showed you another page from the same resource. Does anyone remember what it was? The caffeine one, right? That was from KEGG, K-E-G-G. So in this case, we are visualizing our difference lists through a KEGG diagram. The software knows what genes were found to be differential. Here we are visualizing it using a known pathway because the software has translated our results into gene symbols, and it's able to highlight them within this map to say, these are the genes found to be differential for this particular pathway. It's quite valuable. So if you were to find TGFBR2 to be a significant hit, and you were to find something closely related, say SMAD2 over here, to be closely related, you probably have a greater prior associated with these genes that fall between them. Right? So if two genes that are closely related are found to be differential, the other genes nearby may also have been more likely to be differential. You can always check back through your data to see that. Okay, so these are useful as ways to visualize difference lists. All right. It is also possible to do this in a gene network basis, and we're going to talk about those a little bit in the second half of this talk. So in this case, we see that MMP3, right in the middle of this little cluster, is differential, and you can get a sense of which genes directly interact with MMP3 based on the fact that they have edges directly connecting them from one to the other. You can see in this case that what we have is a hierarchy, and within these hierarchies we have p-values that we've produced as well. In this case, when we look at M musculus module 878, we see that its two genes produced a very low adjusted p-value. The box it depends upon, M musculus module 703 right above it, is also marked as a significant hit. But remember what I said about genes being subgroups of the subsets of the boxes that are higher in the hierarchy? It's quite likely that M musculus module 878 and M musculus module 703 overlap quite a lot in which genes they contain. So the fact that these are both significant does not reinforce the other one, right? They overlap in genes, which means the same evidence is overlapping to produce these significant hits. We see that M. musculus module 703 has 3 genes that are coincident on the list. The module 878 has just 2 genes on the list. So one way to think about that is to say only one additional gene is causing module 703 to look significant on top of what's already seen in 878. So these hierarchies have lots of chances for these matches to lead to significant overrepresentation. All right, so there are lots of reasons why these have some problems. One is that thresholding can be quite arbitrary. Imagine that you have a p-value for each of 10,000 genes from your microarray experiment. And now you've said, I'm only going to accept something with a false discovery rate of 5% or less. So I'm correcting the p-values through Benjamin-Hochberg, and now I've got this list of things, and I'm saying this set of genes is differential, this set of genes is not differential. That's a threshold, and we can see that where I set that threshold will make really big changes in how many genes I claim to be differential. If I accept a false discovery rate up to 10% or down to only 2%, the number of genes I claim to be differential changes a lot. So that's an arbitrariness problem. Within the significant gene lists, we frequently ignore the fact that some of these have minuscule p-values and some of them are right on the line. So if you do that, you're losing information about which ones are the surest hits and which ones are probably differential, but we're not sure. Treating these pathway genes as a set ignores that some genes, like the MMP3 that we were looking at just a moment ago, are quite central in their interactions with others. Other genes are more tangentially related. So some of the things that we've talked about with overrepresentation analysis are actually kind of unsatisfactory. We would like better ways to deal with them. Gene Set Enrichment Analysis is an approach from Jill Messeroff et al. That has a lot to offer us in this space. It's going to remove this hard threshold problem and give us a better way to work with our data than that. So this is kind of their splash, but you can see that Gene Set Enrichment Analysis is available directly from their website. Not to mention being incorporated in lots of other toolkits at this point. So I want you to look at A for the moment. Under A, we see that 2 different cohorts of microarray experiments have been performed, A and B. And from this, we have moved out, we've pulled aside quite a lot of the data. We're not going to look at every gene in this case. Instead, we will probably say, I'm only interested in seeing it in including a gene if it has a variability at least this high. So we can then strip away a whole lot of genes that have so little motion to them that they're not really worth watching. And now we've sorted the genes from top to bottom. Now, I've kind of represented things by saying we typically sort all the low p-values together and all the non-low p-values into this great heap of nothingness. We're stepping aside from that now. We're now adding assignedness to this. So you could imagine that one gene is very high in cohort A and very low in cohort B. In a case like that, it is upregulated and significant. But you could imagine that just the reverse obtains, that it's very high in cohort B but very low in cohort A. In a case like that, we have a gene that we would say is way down and significant. So in effect, our gene ranking here is going to have extreme values that are low p-values and middling values that are much more ambiguous results. So the ups and the downs have been separated in this case. So having sorted the genes in this way, we can then draw a line across this little box everywhere that we have a gene related to a particular pathway, be it development or the serine genes we were looking at just a moment ago. But whatever pathway you're looking at, you simply draw a line across everywhere there's a hit where this particular pathway matches to one of the genes in your list. So what does that look like? If you have a pathway that has a whole bunch of hashes at one end of this box, you think to yourself, that is a really interesting pathway, right? So if it has a whole bunch of lines at the top, that means that the genes that in class A, this is way up. If you have a whole bunch of slashes down here, you make the opposite proposition. You say, this pathway is way up in B, but not in A. So how do you draw that? So here we've turned this graph on its side to throw it up at the top. And what we see is that we have a whole bunch of crossings on that box drawn at the left edge. And every time it hits one of these crossings, it's going to push this value higher. If it adds a gene without increasing that value, it's going to dip back a little bit. So what we see is that these matches lead to this gradual rise of this red line to this point, after which it doesn't really see very many hits to the end of the list. So the further deviated this path is from the 0 line, the more likely it is that this pathway is associated with this list of genes. So this is very nice in that it doesn't require that we look only at the significant hits. Instead, we're able to include marginal information that might give us additional validation that this pathway stands out as enriched. It's become a very commonly used technique in microarray analysis. All right, so in pathways, it's lovely. We have lots of ways to organize our data. Gene Ontology and pathway databases make a lot of this feasible. We have overrepresentation analysis, gene set enrichment analysis, and yet we have a lot of information that's kind of missing here. Not all of the genes for any species have been annotated to a particular function. And if they're missing, that could mean that we're losing some of the information about biology that's taking place in the sample. Welcome back to the second half, where we move away from pathways and start thinking about genetic networks more explicitly. All right, so we're going to start with a little bit of information about graph theory. I think a lot of people don't get a huge background in this, so I don't want to use a bunch of terms that you've never heard before. So we're going to start with nodes. When we create a graph, we have kind of the islands that we're connecting together. Those are the nodes. It's a vertex, generally representing an object or a concept. In the case of genetic networks, it frequently represents a gene or a transcription factor or a protein of some sort. We see that we have edges then that connect these nodes. Those edges represent some known relationship between a pair of nodes. We've already used the term digraph earlier on to describe reactions going in one direction or the other. So if we have edges that have a clear direction to them, then we might have a digraph in place. Some nodes are really, really popular, and some nodes are not. If you have a loner in the classroom, that person may not have a lot of edges to other people. But then there's the very, very popular person who is the subject of, who's just always got something going on and always visiting with any of 15 different people. That's a person who has a high degree, a large number of edges connecting them to other nodes. And we often talk about a connected component, which is to say a set of interconnected nodes that have no edges to nodes outside the set. So if you imagine the group of friends in class who talk to each other but never ever talk to anyone else, that's an example of a connected component. But oh no, I saw that. No hand gestures pointing out to who's who. Yeah, they see you there. All right, so we have this set of a connected component that describes basically what happens if you grab a particular node and pull it out, what comes with it, in other words. So some of the terms that we use can seem a little familiar, things like a clique. So a clique is a set of nodes for which every possible connection is present. One of the smallest examples of this would be, say, 3 nodes, and A is connected to B, A is connected to C, and B is connected to C. That's just a triangle, right? As we move larger, is a square a clique? Point A, point B, point C, point D. A is connected to B, A is connected to C, A is connected to D, right? So we need to have all of those internal connections. B must be connected to D, B must be connected to C, C must be connected to D, etc. So when we create a clique, what we see is that every possible relationship within this set is is present. So in this case, a click of 3 looks like a triangle. A click of 4 looks like a square with a cross in the middle to connect the corners, the opposite corners. All right. We often talk about path length, which is to say if we are trying to go from A to B, how many different edges do we need to travel on the shortest distance to get from one to the other? And we talked about degree just a moment ago. If you have a particular node in the graph that is connected to a very large number of other things, we say that it's a hub. We might also say that it has a high degree of betweenness, which is to say that if you want to get from A to B, you're always going to have to go through D somewhere, through C or D or whatever, a high frequency of the time. So hubs are very, very important genes that play a role in lots and lots of different interactions. All right. So it might be that you're working with a physical interaction network, which is to say that these two genes or gene products must come together physically for this interaction to take place. A lot of the networks that I work with, for example, are protein-protein interaction networks. If you have a case where a protein complex is forming, and without that complex forming, the activity doesn't take place, that's an example of this kind of protein-protein interaction. You might have a signaling network, which could be a little different. What if you have a kinase cascade where the signal that arrives at the cell, maybe EGF arrives, and then that leads to A phosphorylating B, and B phosphorylates C, and C phosphorylates D. That's a signaling network. You might have a gene regulatory network. Often these are graphs that involve multiple kinds of shapes. We have an example over here at the right. You might represent transcription factors as circles and their targets as boxes. And so these edges represent that this transcription factor stimulates all of these genes. That's a gene regulatory network. Metabolic networks, we've already looked at examples. You saw caffeine earlier. That's an example of gene products that all follow each other in a concrete metabolomic pathway. Co-expression networks describe a likely link in function, which is to say, under the same stimuli, these genes show similar patterns of expression, that when A goes up, B goes up. When A goes down, B goes down. Co-expression. And genetic networks. You can have all kinds of ways in which genes have a strong relationship on which other genes are being expressed. Genetic networks. Okay. There are 3 principles that I would really, really like you to absorb about biological networks. Scale-free, small world, and hierarchical modular. I don't want you to be thrown off by these terms because they show up in a lot of places and they describe some of the complexities of how we operate. Has everyone read some sort of paper where one of these big spiky hairballs shows up? Okay, well it's a favorite kind of thing to do in a systems biology paper. They are not actually that informative. But these real-world relationships among genes all can be broken down to these 3 basic concepts that I think everyone should get. First is to say biological networks are scale-free, which is to say that there are a few genes that operate as hubs, and most genes have a relatively low degree associated with them. So if I am evaluating how interactions take place among different gene products, it's very frequently the case that a hub is going to mediate the interaction of multiple genes. Does everyone remember the 6 Degrees of Kevin Bacon? That's a little old at this point.
Kevin Bacon, what?
Kevin Bacon is the actor. Kevin Bacon has been in a surprising number of films over his career. And so if you're trying to ask, how are these 2 actors related, you might frequently come to a place where you say, well, this person was in a movie with Kevin Bacon, and this guy was in a movie with Kevin Bacon. Therefore, they're related by 2 degrees of Kevin Bacon. Right? So this is a principle that is just as relevant in human gene networks. If you're asking, how do these 2 genes relate, you can ask, how many gene partners do you have to go through to get from A to B? Typically, in something like humans, you see something like 6 degree separation, that there's not a huge number of steps that you need to go in order to get from gene A to gene B, so long as you know something about what their function is. That you can almost always find some pathway through known genes to get from one to the other that is not a huge number of steps. So we have a highly interactive set of gene relationships within our species and others. And these hubs are one of the reasons why that's true, because a very small number of genes that are hugely interactive with others is a big deal. I missed telling you something about scale-free networks that's worth repeating. If I choose one of 20,000 human genes at random and I knock that puppy down, how likely am I to create a cell line that isn't viable, that can't grow? If I choose a random gene in a human.
Not very likely.
Right. Most of the genes that you just take out randomly from the human genome would not cause the resulting cells to grow, to be killed. I mean, our genome can tolerate some number of its genes being inactive and still grow. If, however, you take away, if you knock down a gene that has that happens to be a hub, you're in a world of hurt. So a very few genes play a massively important role. They are not, our cells are not tolerant to losing the function of that gene. In the main, most of our genes, on the other hand, are not essential. So we have in a lot of cases sort of redundant mechanisms, that if this pathway to get there is not working, there's another route to get there. Scale-free is kind of where that comes in. This last concept, I think, can be a little harder to get. So I'm going to try to just do this visually. If we were to look at this set of 4 genes, these little red ones right here, we would see that those 4 are all completely interconnected. They are a clique in this case. So we could call these a module, just this tiny little set of 4. At a higher level, to make a hierarchy, we see that collectively, this set of genes, which now includes 16 genes, are also a module. They're very, very tightly related to each other. We see that this set of 4 and this set of 4, they're internally very highly connected. These create a connected component within them. When we pull together all 16 of those, they're also very, very much more tightly connected internally than to anything externally. And then at a higher level yet, we see that this set of genes and this set of genes and this set of genes and the others within it, they are as well connected very tightly internally. So what we see is that at a very small level, with very small numbers of genes, we frequently see modules, completely connected components within them, very tightly connected components, operating as a set. And at the higher level, that set is part of another set that tends to work together very constructively. So if you're trying to understand what relationships look like within the gene sets that we see in our organisms, we see that scale-free principles, small world principles, and hierarchical modular principles are all at work in how we find genes operating together. This paper from 2004 might be a little old, but the lessons from it have stood up really well. All right. There are a world of tools out there for visualizing networks. Here I've included links for 6 of them. I would just note that the elephant in the room is Cytoscape. For this, I would ask you to return to the days when Dave Tabb was in grad school. I went to grad school in 1996 at the University of Washington. I'd never lived on the west coast of the States before. I was kind of terrified. And when I arrived at the University of Washington, I learned that the other student who had been brought in, this was the first time this department was bringing students from the outside world, the other student was this interesting guy named Trey. So I was used to being a very big fish in my little state school. I was a top scholar as an undergrad, and when I went to grad school, I thought, this is going to be nothing. I prepared all my life to go to grad school, but I got to tell you, I felt like a big bozo for much of the first half of the time I was in grad school. And one of the reasons was that my classmate, Trey, was amazing. The guy already had his master's degree from Massachusetts Institute of Technology. He had a clear vision of what he wanted to do with his career. He was just blowing through the classes that we took together, and I was like, am I a loser? What's going on here? Trey Eidecker went on to create a lab very shortly after graduating. He became a professor almost right away at University of California, San Diego, and he created this Cytoscape software. So my inferiority complex continues to this day. 2 decades later, I'm still like, who was this guy? I just want to say, if I can make it through the abject depression I spent my first couple of years of PhD work in, you can too. You can too. And I think I did okay in the end, but to see him excel in the way that he always has been challenging to my self-worth. All right, so Cytoscape is an amazing toolkit for visualizing all kinds of gene-gene, protein-protein, transcription factor-to-gene relationships. It's amazing. A beautiful tool. I would definitely recommend that you know something about it because you're likely to use it to visualize your data at some point in your research. All right. So it is able to integrate and visualize data through networks. We often think of proteomics data as existing in conjunction with more proteomics data, and genetic data as relating only to genetic data, and transcriptomes, well, we kind of project them at the level of gene, etc. But you can actually combine across many of these different data types and visualize them in one coherent way using Cytoscape. So it is definitely the big story of visualization within this genetic network space. All right. We have mentioned the small world aspect of biological networks, which is to say that if I have a gene A and B drawn from within the genome, there's probably some relatively small number of steps to get me from one to the other. People have found, though, that when we look at gene-gene networks, we find that there's a high degree of relationship in function that describes how close together genes are. So if you imagine a case where these two genes, A and B, are known to interact directly, if you have functional information on one, you can implicitly learn what function the other is also taking. That if two genes directly interact, there's quite a great deal of functional overlap between the two. Now, if gene A and gene B are separated by a third gene, gene C, there's quite a fall-off in how much relationship the two have in function. And if gene A and B are separated by C and D genes, that you have to relate A to C, C to D, D to B, now the amount of functional relationship between the two genes is quite a lot lower. So we use this walking distance, basically, to get a sense about the functional relatedness of genes to each other. And this paper kind of tries to talk about how we walk through all of the data in functional association with genes to learn about that. All right. It is quite. We talked about hierarchical modular, right? There's an approach that's been put together called NetSam for figuring out ways to tease out modular relationships from these networks. If you imagine that in this case we have the full set of genes in this top diagram, we find that we can separate the blue and green set from those that are in, it's not burgundy, what color would you call that? Lilac? I don't know, fuchsia. Fuchsia dots and orange dots and red dots. We could see that as a first step, we could be divisive to separate the left blob from the right blob. Now within that green section that we've pulled off, we see that we have two different modules represented, the green dots and the blue-green dots, right? So we could pull those apart. This is an example of the hierarchical modular nature of genes. And this gives us a way to organize our data a little more concretely. This is some software called NetGestalt. It's kind of the big brother of the WebGestalt software. And it gives us the ability to not sort the genes alphabetically or cytogenetically, where they appear on the chromosomes, but rather to link together, to bring together genes that are most similar in function on one axis. And the software allows us to visualize microarray data, genetic variation data, and proteomics data all on one axis like this. So in this case, we are visualizing the raw data from a bunch of microarrays in this area. Here, we're doing a visualization of the p-values that were produced for individual genes. Up here we have the gene names to which those data have been associated, and up at the top we see kind of an overlay that reflects how closely related is this block of genes. So it's a very powerful visualization tool, and certainly if you're working in anything human, this is a really great toolkit to work with. Lots of pieces today, and I really appreciate your patience as we got through this. So building your assessment based on pathways or networks is likely to give you a much better appraisal of what you've got than a laundry list. Right? If you go to the grocery store and you have a list of 50 items you're trying to buy, that's complex. But if you know, okay, this set relates to cake, this set relates to dinner tonight, this set is for the bride on the weekend, suddenly it's a little easier to organize it in your head, and you have a less chance of forgetting something important while you're at the grocery store. In the same way, being able to organize your findings as a set of pathways that's differential, a set of gene networks that are differential, network modules that are differential, you have a better chance of being able to interpret and organize your data in a meaningful way. Biological pathways are built on a categorical basis. Something is on the list or it's not on that list. Typically, biological networks tend to be more point-to-point in nature, that we know this pair of genes interact, and one of those is also associated in an interaction with another gene, therefore they form one connected component. Gene Set Enrichment Analysis and overrepresentation analysis very frequently show up when people are using software like relationships like Gene Ontology or Wiki Pathways or something like that, or KEGG, to represent their data. So GSEA and ORA are both important approaches to know about because you'll probably read a paper involving them. Okay, that is our story about biological pathways and networks.